Wonderful. Well, thank you so much for coming to our presentation today. Our online programs, and specifically our Imaging Sciences program, is excited to partner with Dr. Tinker and these wonderful speakers today to discuss COVID vaccines. So I want to introduce Dr. Tinker, who will be our first speaker today. Dr. Tinker is a faculty member of the Department of Biological Sciences in the Biomolecular Graduate Program at Boise State University. She has a PhD in microbiology from the University of Iowa and completed postdoctoral training in bacterial pathogenesis at the University of Colorado before coming to Boise State in 2005. Dr. Tinker's research is on vaccine development, and she also teaches microbiology, microbial pathogenesis, and vaccinology courses, and regularly works within the community to promote vaccine education and public transparency of vaccine science. With that being said, here's Dr. Tinker.
Thank you. Thank you so much, Tom, for that introduction. And thank you, Allison, for having us here today from Extended Studies. And thank you all for coming. It's definitely my honor and privilege to be able to introduce this talented group of students today. They are taking a course at Boise State on COVID-19 vaccines this semester. And I know they're really excited to share all their knowledge with you. I think one of the reasons I was able to convince them today to come and talk is that they all feel that this is really important. And they feel like it's really important to be as transparent as we can be about the science of vaccines. So I think that's why we're here today. So this is kind of the agenda that we're going to cover. It's going to be kind of broken into three groups. So we'll have an intro and I'm going to do a brief intro on vaccines, and then we're going to cover a little bit of the history of the vaccines, the COVID-19 vaccines, some human clinical trial information. The middle of the webinar will be covering the vaccines themselves that have been approved, as well as a new one that's coming shortly. And then at the end, we'll kind of end with some information about the ingredients and kind of more of a global perspective. So we are going to move kind of fast today because we really want to be able to have time for questions at the end of the webinar today. We're going to be collecting questions in the chat room. Hopefully we'll be able to answer those at the end. So I'm going to start here with kind of a broad Vaccine 101 introduction. And I'm mostly introducing some terminology that the students are going to mention later on. So I want to make sure that we get the terms out there. And of course, the first one is vaccination itself. And what is vaccination? And it's important to distinguish vaccination from a therapy or a drug. And that is because vaccines introduce a material that then induces your immune response. So it is actually your own immune response that's triggering the protection against the disease. And sometimes the vaccine is referred to as antigenic material or an antigen. That's a foreign material that your body hasn't seen before to induce the immune response. So what kind of immune response do we need with a vaccine? Well, this is a virus, and viruses tend to induce two types of immune responses. And you're going to hear this term during this webinar, and that's the spike protein. That's a large protein present on the surface of coronavirus that is the antigen that really induces our immune response. Viruses replicate inside of our body, but they also replicate inside of our cells, and that triggers two different types of responses, a T cell response and a B cell response. And a T cell response tends to induce immunity that is going to protect you against things inside of your cells. And a B cell response induces immunity that protects you against things outside of your cells. And B cells produce antibodies. These are big proteins in your blood, and you may have heard of those before because we tend to use those as sort of a goalpost. That's what we're going to detect to identify a good vaccine. So if I was going to give the history of vaccines in one slide, it would be this slide. And it's showing you 15 different diseases that we protect against currently in the United States and globally. And these diseases are mostly childhood diseases that cause lots of mortality. On the left side in the orange, we have the number of cases of each of those diseases before the vaccine. Then on the right, the number of cases after the vaccine. And importantly, I want to point out smallpox here at the bottom. Oops, one other way. Okay. Well, smallpox is actually eradicated globally from the world because of vaccine, and it was considered the most most horrific infectious disease in history. So to categorize vaccines, mainly we classify those into two main traditional classifications, those that are live attenuated. Those vaccines contain live organisms, either bacteria or viruses, that can replicate maybe a little bit, but they cause a much less severe form of disease or no disease at all, and that induces an immune response. We can loosely put the COVID-19 viral vector diseases into this category. That would be the Johnson & Johnson or adenovirus vaccine. Those are live viruses, but they actually can't replicate, so they can't cause disease. Inactivated vaccines then are going to be killed forms of viruses or bacteria or parts of those that are purified. We have polio, tetanus, meningitis, and also influenza in this group. And loosely, we can put the COVID-19 mRNA vaccines into this group because they're killed as just parts of the virus. So we're going to be talking about mRNA during this webinar. So I'm going to step back and do just a brief overview of how the flow of genetic information occurs inside of a cell. And many of you guys probably already know this information, but you're going to hear it again today. So on the left side of the slide, kind of underneath the half circle, are things that happen inside of the nucleus of a cell near the chromosome. And the chromosome is made out of DNA, so it's going to go through replication to make more of itself. Inside the nucleus, also, we have transcription, which takes the DNA and turns it into RNA, or messenger RNA, which gets transferred outside of the nucleus of a cell, and there it goes through translation to make protein. So those are some of the basic steps that you guys are going to be hearing about with our vaccines. And I'm now going to hand it off to Leslie Kendrick. Leslie Kendrick is an associate professor of radiologic sciences at Boise State and also the director of imaging sciences. And she's also a graduate student in the biomolecular PhD program. So.
Thank you, Dr. Tinker. I'm going to spend just a few minutes talking about the history of coronavirus and then just a bit about the different types of COVID-19 vaccines that are in development. This is really touchy. There we go. So historically, there have been four different types of a vaccine, four different types of, oh my goodness, I might have to give this to you, Elise, because I'm not even touching it and it's scrolling back. Elise, do you want to take it? And I'll just say next slide. Okay, perfect. Thank you. All right. So historically, there have been four types of coronavirus that humans have been particularly susceptible to. They've generally caused very minor signs and symptoms, and our immune systems have taken care of them pretty easily. In the last 20 years, we have had three new coronaviruses that have popped up. And so we'll talk just a bit about those. In 2002, we had SARS. It was identified and it spread across 29 countries. It has been recorded as causing just over 8,000 cases and led to 774 recorded deaths. Then we had in 2012, MERS was identified. It had a second outbreak in 2015 and a third in 2018, and was known to spread across 27 countries. And as of last year, was reported to have caused just over 2,500 cases and lead to 866 deaths. Then in 2019, we had SARS-CoV-2, which is the coronavirus responsible for creating the disease COVID-19. It has since spread over 219 countries and territories and has led to over 128.7 million cases and unfortunately more than 2.8 million deaths. Next slide. So if you look at what these three different viruses have in common, they do have four proteins at the end of their 3' end, which is the end that's responsible for entering into the human cell. In common, they have the S, the E, M, and N, which stand for the spike, envelope, membrane, and nucleocapsid protein. If you look at the bottom down here, they're all pretty, this is a pretty simplistic presentation of picture of that end, but they're all pretty conserved in the way that they are aligned with each other. And for that reason, researchers have really focused on these 4 proteins for the development of vaccines for these 3 viruses. And specifically because the spike protein sticks out a little bit farther. There's the nucleocapsid that's encapsulated by the membrane and the envelope proteins, but the spike protein, like Dr. Tinker said, sticks out a little bit further. And for that reason, it is the one that attaches onto the human cell with the receptor binding domain and is a great focus for the development of SARS-CoV-2 vaccines. Next slide. Dr. Tinker did talk about how there are live attenuated and inactivated as far as categories of different types of vaccines. So I want to break those out into a little bit of subcategories, just so we have a further understanding of the different subcategories of these 2 main categories. So across the globe, there is more than 45 different COVID-19 vaccines that are in phase 2 and 3 trials. These vaccines all have a common goal. Like Dr. Tinker said, they are attempting to initiate the immune response to the SARS-CoV-2 virus so that it prevents or eliminates the development of the COVID-19 disease, or it keeps it from progressing to a significant disease process. So within that, we have each one of those vaccines is one of 7 different types. So if we look at the top underneath the live attenuated, we see that there's a viral vector. A viral vector vaccine uses a harmless virus to house a piece of the virus of interest and uses that as a vehicle to carry that protein into the individual. A live attenuated virus vaccine is one where you actually take the full virus and you eliminate the most infectious pieces or components associated with that virus before it's developed into a vaccine. When you look over on the inactivated portion of the types of vaccines, we have the RNA, and the RNA uses mRNA, which is actually a component of messenger RNA. It's just a piece of the protein instructions so that those instructions will get into the individual and produce the protein to initiate an immune response. And that is housed within a lipid shell or a liposome. The Pfizer and Moderna are this type of a vaccine, and those are the 2 that are available in this category for emergency use in the United States. When we look at the other remaining types of inactivated, we have the inactivated virus that is associated with a whole virus again. It has been chemically modified to eliminate its ability to replicate, but it initiates an immune response. Then we have a particle subunit or subunit viral protein vaccine, which is a piece of the protein or a whole protein that is added with an adjuvant. An adjuvant is an immune activating material that will initiate the immune response against that particular protein. And then there is the like virus, the virus-like particle type of vaccine, and that is where you take an assembled proteins that are like the virus that are incorporated in an adjuvant to develop a vaccine. And lastly, there is the DNA vaccine, which is the genome of those particular proteins of interest, but they don't have any of the replicating material within that vaccine. So the Johnson & Johnson is the viral vector type of a vaccine, like Dr. Tinker mentioned, but there's also another vaccine that is going to be out hopefully within the next month for emergency use, which is the Novavax vaccine. And that is a subunit viral protein vaccine that will help hopefully with the availability of vaccines within the United States within the next month as well. So we will talk about each one of those throughout the rest of our presentation. And with that, I'd like to turn this over to our next presenter, which is Elise Overgaard. Elise is a PhD student in our biomolecular sciences program here at Boise State University.
Thanks, Leslie. I'm going to give a brief overview of human clinical trials and try to touch on some of the questions about why and how these COVID-19 vaccine trials are different from normal vaccine trials. So I want to start with an overview of how a vaccine is normally discovered and brought to market. I'm going to say vaccines, but all drugs go through the same process. In my diagram here, you can see it's basically an inverted pyramid process. We start with thousands of potential compounds and eventually whittle that down to a handful of approved vaccines. At the base of the pyramid is what we call basic research. Scientists here are really focused on exploring a broad problem as opposed to specifically bringing a drug to market. At this level, scientists might be studying the genetics of the virus or like how a specific viral protein works. The next level is actually drug discovery, and that's more targeted. That's where the scientists start actively trying to solve the problem. They ask, how can we utilize that virus's genetics, or how can we utilize that viral protein to actually create a vaccine? And they figure out how to actually make the compound and how to deliver it into humans. And then promising ideas from that stage move into preclinical research, where they actually start to test the vaccine in animal models. Once a compound is shown to be safe in animals and that it can elicit a protective immune response, then the company or the academic institution can apply, can put in an investigational new drug application with the FDA. This IND application has to be approved before they can ever test the compound in humans. The FDA reviews all of the data that comes out of the basic research, drug discovery, and preclinical phases. It's a huge body of research, and there has to be a really good reason to test a new compound in humans. So once the FDA grants permission, then we can start testing in humans. We call that phase clinical research. So this encompasses all of the human testing. And once all of the human testing is complete, then the company or the institution files a biologics license application with the FDA. And all of the human data gets reviewed again by the FDA before they make the decision to license. So with these COVID vaccines, we really lucked out and had a major body of basic research and drug discovery and preclinical research already completed. We literally had decades of work that scientists have been doing on those SARS and MERS viruses, like Leslie mentioned. And there was already a huge body of background work done on these platforms that kind of seem like they popped up out of nowhere, like the mRNA and adenovirus platforms. We haven't seen them in practice before, but in reality, scientists have been working on those platforms for literal decades. So we hit the timing just right to be able to push those new platforms over the edge into clinical use with the pandemic here. So it wasn't like we were starting from the base of the pyramid. Another thing is that because of the pressure of the pandemic, the FDA has been able to prioritize all of these COVID vaccine INDs, the investigational new drug applications. And so they were literally just able, like physically, to push the paperwork through faster than they normally would. I want to quickly dive into some specifics about how human clinical trials work. I also want to point out, keep in mind as we go that the patients who we test new compounds on, they're all volunteers. They're all just normal people with normal careers and families. They take time out of their own schedules to show up to clinics and provide data and blood samples and whatever else the companies need to figure out if the vaccine works and if it's safe. So normally we start with a phase 1 trial to confirm that the vaccine is safe. We test it just in a handful of people, literally a dozen or 20, and we make certain that there's no obvious safety concerns. Once we know it's safe in normal healthy patients, then we can move on to phase 2. In phase 2, we test it in more people, maybe a few hundred, and try and answer the question, do we actually make an immune response to the vaccine? We keep looking for safety here, always safety. So if we can show that the vaccine does make an immune response in phase 2, then we can move on to phase 3. In phase 3, we start saying, great, we made an immune response, but does the immune response actually protect you from the disease? So we start to look for vaccine efficacy here. Normally we have to have a complete data set from each phase in order to move on to the next phase. But what's happening with COVID vaccines is that they've been able to combine like phase 1 and phase 2 together. So they can test it in a few patients to show it's safe, but then instead of waiting to write up all of the results and submit those for review and then apply for phase 2, they're able to just roll straight into the phase 2 studies. And the same thing is happening between phase 2 and phase 3. Instead of waiting to write up all of the results, they're able to roll straight from phase 2 into phase 3, which saves a lot of time. There are a lot of safety checks in place to make sure that these aren't moving too fast. There are literally hundreds of sets of eyes on this data, and there are really hard qualitative stop rules in place to prevent a vaccine from moving on if there are concerns about safety. I'm going to talk about those in a minute, but the other thing is the FDA has a lot of data. They have all hands on deck right now reviewing this data the second it becomes available. So these aren't just sitting around waiting for somebody to take a look at them. Another thing I want to point out is that what's happening with COVID vaccines right now is emergency use authorization. So these vaccines are not actually licensed yet. Normally, they can't license a vaccine until all of the safety and efficacy data from all of the phases is submitted to the FDA. So right now we're just looking at emergency use authorizations. And I'm going to go over that in a minute too. But before I leave this slide, I just want to point out that because we're only in emergency use authorization mode, a lot of these late phase trials are still ongoing, and they are depending on patients who are in the studies to stay in the studies, which is becoming harder as the vaccines become authorized and they're available in people's communities. It's harder to keep patients in the studies if a vaccine is available. So that's a conversation for another time, but I wanted to draw some attention to it that these are still ongoing. Okay, so I want to go over how adverse events get reported in clinical trials, which is different than how they get reported after the clinical trials. Rachel will talk to you a little bit about that. During clinical trials, there are certain things that we know will happen with the vaccination. This is some data from the Novavax trial, phase 1 trial. I'm just going to use it as an example. So the things that we know will happen or expect to happen, we call them solicited adverse events. They're events that are solicited. They actually ask the patients, did you experience any of these things? So some of those things are local at the injection site, like redness or swelling or pain or tenderness. If you have time to look at these slides, you can see the placebo group is included here. So they are collecting this on the placebo as well as the treatment groups. The other kinds of solicited adverse events they look for are systemic things. So anything like arthralgia, which is joint pain, fatigue, fever, headache, myalgia, which is muscle pain, nausea, malaise. These are all things they kind of expect to happen with vaccination. So they ask patients, are you experiencing these? Everything else still gets recorded. So any change from baseline, any change from a patient's baseline gets recorded as an unsolicited adverse event. That can include anything from getting food poisoning to breaking your ankle, anything at all, anything that's different from a patient's normal gets reported. They also have some categories of things that would trigger a stop rules or at least a look at the data, and those are serious adverse events, which include death, hospitalization, life-threatening illnesses, disability or permanent damage, and congenital anomalies or birth defects. Any of those are considered serious, and they warrant a deeper look at what's going on. They also record adverse events of special interest, which is any COVID-19 related complication, and then any immune-mediated medical conditions, which is just anything involving the immune system that could have been triggered by the vaccine. All of these things require like a stop and a look at the data to see what's going on. Okay, the last thing I want to touch on with my time is how emergency use authorizations happen. These are basically a way to allow a vaccine or a drug to become available during an emergency situation where there's no alternative treatment or therapy. So first of all, first question, who are the people who are deciding if these brand new vaccines are okay for everyone to start using? Well, the people who make the final call are the FDA's career scientists and physicians. These are people who have a lot of experience in reviewing scientific data and in making public health decisions. But they don't just decide on their own. Before they make the call, there's a public advisory committee meeting, and this consists of dozens and dozens of external non-FDA scientists, doctors, public health experts, people from all over the country. They basically review the data and testify for or against the vaccine. These meetings can last 8 to 10 hours, maybe longer. And then the FDA committee takes all of that input and they make the final decision. So the important thing I want to point out is that everyone involved in these decisions are people. They all have families and careers, they have children, they have parents. These people are independent of drug companies, there's nothing in it for them, and they have as much interest in keeping unsafe things off the market as everybody else. Okay, so that's who's deciding, but what do they base their decision on? They basically have to prove that the known and potential benefits of the vaccine outweigh the known and potential risks of the vaccine. They look at the chemistry of the compound, the manufacturing, all of the safety controls that are in place to make sure that the vaccine is being made with high quality and high consistency. And then they also consider whatever safety and efficacy data is available at that point. So I want to touch on what that entails, what safety and efficacy data actually means, what they're actually looking at. They have to have the complete safety data from phase 1 and 2. They can't skimp on that. There's all of the safety data has to be submitted from phase 1 and 2. And then for phase 3, they still have to have data. They have to have some efficacy data, and that has to meet an endpoint that they have specified before the trial even starts. They also have to have followed up at least half of the people who got vaccine for at least 2 months and have shown that there's no safety concerns. And they also have to have at least 3,000 people vaccinated, and those 3,000 people have to be followed for at least 1 month for safety events. So the decision is really based on a pretty comprehensive data set, and they're not skimping on safety, I guess, is the end point. So that's my piece. I also wanted to point out that all of the phase 3 protocols for these vaccines you're going to hear about next, these are all available online. So you can go check them out yourself and see what kind of data they're collecting. Okay, so I'm going to hand it over to Sierra. She is in the B2B biology program, and Sierra, it is all yours.
Thanks, Elise. Okay. So thanks, everyone. Hi, I'm Sierra, and it's my pleasure to speak to you all about the fabulous technology of mRNA vaccines against COVID-19. Okay, so I wanted to open up this presentation with some context on this technology. The only thing that is new here is the vaccine themselves. And this technology has actually been around for over 30 years now, with a strong interest in cancer and infectious disease treatment. And so, in fact, it's actually mentioned that mRNA is actually a safer vaccine alternative, as this molecule, it does not cause infection, it degrades quickly in the body, and it doesn't have the hardware to integrate itself into our DNA or have the potential to cause harmful infections. And so I also have here a timeline, oops, too fast, a timeline kind of giving us a visual representation of the time and research put into oh, too fast now, gosh. Okay, here we go. Maybe we'll skip that, that's okay. And so some questions that you might be asking yourselves is, will this vaccine make me sick? And will this vaccine affect my DNA? And the short answer for both is no, but I'll go into a little bit more detail in a moment. But first, just a short video on a very general overview on how these mRNA vaccines work. Oh no, maybe we can't hear the sound. Well, I'm not sure if we could hear that. Oh, there it goes.
This is the little protein that COVID rides around on. And you get the vaccine, you're not getting this or this. You're actually getting this instructions in the form of mRNA on how to make this.
Perfect. Little technical difficulty, but that's okay. We're moving on, maybe. Perfect. Okay. So this infographic is available online, cdc.gov to be specific, and it does go into a little bit more detail, but for presentation purposes, I did simplify a bit. And so to put it into a little bit more perspective, this vaccine is essentially providing your own immune system with the knowledge it needs for a potential fight against COVID-19. So we're working smarter, not harder. And so the card was representing the spike protein here. And then we also have the instructions provided to our cells in the form of mRNA. The package here represents the mRNA delivered to our cells. And to answer the question of, will this vaccine change my DNA? No, the delivery of mRNA to our cells does not impact our DNA at all. The mRNA will not encounter the cellular structures that perform DNA replication. And so again, the present here, it represents the packaged mRNA molecule that's delivered within a lipid nanoparticle. And then once the vaccine is administered to our cells, and then our cells receive that information in the form of mRNA, it can begin to produce antibodies that will recognize and attack anything that resembles the spike protein. So now you might begin to feel a little bit crappy after your 2 doses of vaccine. And to answer the question of, will this vaccine make me sick? There is no infectious agent within the mRNA, but the side effects you might experience, like headache or fatigue, that's your body beginning to manufacture those antibodies that will recognize the spike protein should you be exposed to the virus. And so some takeaways I want to leave you with. Pfizer and Moderna are the current mRNA vaccines on the market, each showing a greater than 90% effectiveness or protection against COVID-19. So these mRNA vaccines are well-researched, safe, and effective. They will not encounter the structures that perform DNA replication. And these mRNA vaccines, they allow our bodies to work smarter, not harder, by using genetic information to produce antibodies against COVID-19. And with that, I'd like to pass the virtual baton to Haley. Haley Bridgewater, she's a PhD student, also in the biomolecular program.
Thank you, Sierra. Hello, everyone. Thank you for attending our webinar today. And I'm going to be talking to you about the AD.26.cov2.S vaccine, more commonly known as the Johnson & Johnson or the Janssen vaccine. AD26 is the adenovirus vector that's used in this vaccine, and I'll be talking a little bit more about that in a moment. And CoV2 stands for the SARS-CoV-2, the virus that it's protecting against, and I'm sure you're familiar with that. And the S stands for the spike protein that you've heard everyone else mention is on the surface of the coronavirus. And it is the way our body will identify the virus once we've been vaccinated. All right. So recently, the Johnson & Johnson vaccine was granted emergency use authorization by the FDA, meaning that it can now be publicly distributed. And in the letter of authorization, it was stated that the vaccine is a recombinant, replication incompetent human adenovirus serotype 26 vector encoding the SARS-CoV-2 viral spike S glycoprotein. So that's super complicated, and that's what was released to the public. So let's break it down a little bit so we know exactly what's happening in this vaccine. So as I mentioned earlier, the Johnson & Johnson vaccine uses an adenovirus 26 vector. AD26 is a virus that infects humans and will present with common cold symptoms. They chose this virus because they've used it in other vaccines like the Ebola vaccine, and so it has an established safety record. And you may be asking yourself, if it causes the common cold symptoms, will I be getting sick from this vaccine? And that is where the recombinant and replication incompetent portion of the statement comes in. In the laboratory, they cut out a chunk of the adenovirus genome, and that prevents it from producing new viral particles or from mutating. So the adenovirus vector will not make you sick when you get this vaccine. The next part of the statement is encoding for the SARS-CoV-2 viral spike S glycoprotein. This means that the genes for the S or the SARS-CoV-2 S or the spike protein is put into the adenovirus vector. And then once the adenovirus vector sneaks into our cells can produce the spike protein of the SARS-CoV-2 virus. Our immune system will then recognize that individual protein as something that's foreign, and it will produce antibodies against SARS-CoV-2. And then a portion of those antibodies will be stored for later, so that if our body encounters any future infections by SARS-CoV-2, we will be able to prevent them. All right. Now that you know a little bit more about how the Johnson & Johnson vaccine works, I want to talk about efficacy and safety. I've heard a lot of people saying that they don't want this vaccine because it was rushed and it's not safe, or that it has less efficacy than the mRNA vaccines. So hopefully I'll give you in this talk a chance to think about those. So as Elise mentioned earlier, phase 3 trials are still ongoing, so we don't have the extensive long-term safety data, but the FDA is being regularly updated, and they have released no safety concerns with the Johnson & Johnson vaccine. Additionally, they released all of the phase 2 trial data, and I went through it, and here's a little of the highlights from that. They vaccinated 405 individuals who were 18 or older, and of those 405 vaccinated, they only had one severe side effect, and it was a fever, and the person who had that sought medical attention. And that fever was resolved within 12 hours, and it had no lasting impacts on that individual. So overall, the safety data for Johnson & Johnson vaccine looks very positive. Another concern I've heard about the vaccine is related to efficacy. So let's talk a little bit about that. So the FDA has reported the efficacy to be 66.9% for this vaccine. And vaccine efficacy means that the vaccinated group in the clinical trial had a 66.9% reduction in disease compared to the control or non-vaccinated group. And this is lower than the mRNA vaccines for COVID-19, but by getting this vaccine, you are still greatly reducing your risk of getting the virus, and you are reducing your risk of a severe infection if you do get sick. Also, just a quick reference in relation to other vaccines, the yearly flu vaccine tends to be about 20% less effective than this, and it still reduces disease. All right, so while the efficacy may be lower than an mRNA vaccine, there are key advantages to this vaccine, mainly that you only need one shot to achieve a long-term immunity, which can drastically increase the number of people that can be vaccinated in a short period of time. Additionally, the storage temperature is that of a normal refrigerator, or 4 degrees C, which can allow vaccines to reach more rural and resource-limited places. So as someone who is in this field, I would happily take this vaccine to protect myself and those around me. And additionally, having an increased number of people vaccinated per week will help us to end this pandemic much faster. So thank you for listening to my portion of this talk. And I'm going to pass it over now to Tana Gazdick, who is another PhD student in the Biomolecular Sciences program. Thank you for listening.
Thank you, Haley. As she mentioned, I'm going to briefly introduce Novavax, which is a spike protein vaccine. It does not currently have emergency FDA approval, but they are hoping to have that by May. So very soon. Okay, so what is it made of? So the two major components are the spike protein nanoparticles, and then also the matrix M adjuvant. In this case, they use saponin, which is extracted from the Chilean soapbark tree. This picture represents that right there. And interestingly, we already use it in food products such as root beer, foam, and Slurpees. So we already know it's safe to use. But there is a problem with creating vaccines for the spike protein. The major problem is that this form right here to the left, it's called the prefusion form. It doesn't want to stay like that. It actually would like to stay in this post-fusion form. And what this means is the prefusion is the form it is before the virus infects our cells. And so we want to create a vaccine towards this form because we want the vaccine to destroy the virus before we're infected. And so past research has been done, and they found that proline substitutions at these two locations, which is basically just changing two little tiny parts of this protein, they found that this stabilizes it in this form for both MERS and SARS-1. And so scientists were able to take that information and see if it works on COVID, and it does. And so that's just another example of how all this research has already been done on all of this, and we're able to apply it to current situations. Okay. So how is it made? Well, first we need a lot of the spike protein. And so instead of doing it ourselves, we use other things to make it easier for us. So we take the DNA and we put it into moth cells. And these moth cells do all our work for us, and they make a whole bunch of this spike protein. And we're able to take all that spike protein and purify it from those moth cells so that there's no other part of the cells in there, just the protein. And then we can put it onto these nanoparticles. And so you have all these spike proteins sticking out from the nanoparticles ready for your immune system to interact with it. And then you take that and you add it to your adjuvant, your saponin, and then you have the vaccine. So it's super simple. And then you can give it to patients. Okay, so the next part, I really wanted to show you what phase 1 and 2 trial results looked like. And I know it can be a little bit confusing, but I will walk you through it. So for this y-axis, you have your levels of antibody in response to the spike protein. And the higher up these dots are, that means the more antibody that was detected for that. And then for your x-axis, you have the different test groups. And they were all tested over the course of 35 days with 2 doses each. And so, let's see if it goes. Nope. Maybe. It's not working. Okay. Oh, there we go. So for our first test group, they were given placebos for both 1 and 2 doses. And the placebo in this case is just saline. And as you can see, there isn't really much of a response. Most of the data is down here, barely any levels at all, which is what should be expected to see. Then for our next group, they were just given the spike protein and not the adjuvant. And you can see that there was some antibody response after the second dose, but it's still not at the levels we want it to be at. And so for the next 2 groups, we have both the spike protein and the adjuvant for both doses. But another thing they're testing here is the concentration that they need to have this be effective. And so they tested 5 micrograms versus 25 micrograms. And you can see for these results, they look great. And the important thing to note is that there isn't really a difference between the 5 and the 25 micrograms. So this means they can get away with using a less concentrated vaccine dose, which means more vaccines for more people. So it's good news. Okay. And the last group, for the first dose, they have the spike protein and the adjuvant, but then they gave the placebo for the second dose. And you can see that after the placebo is given, there wasn't any change. So this means that 2 doses are required. 1 dose isn't enough to get those high levels of antibodies. And so for this last figure, it says it's the antibody level versus human convalescent serum. And this basically means people that were naturally infected with COVID were able to donate their serum to be tested. And so if you can see this blue line right here, it's comparing people who had little or no symptoms during infection. You can see their antibody levels are pretty low. And then if you go to people who had severe enough COVID that they had to be hospitalized, you can see that they're about equal or even a little bit less than people who had both doses of the vaccine. So my takeaway message for this is that it is most likely better to get the vaccine versus getting COVID naturally, because we don't want to be hospitalized just to get that level of antibodies, right? And as far as their short-term safety goes, there were no serious adverse events recorded as of yet, and just regular vaccine symptoms like sore arms and slight increased temperatures and headaches. Okay. So yeah, next I will hand it over to Gianluca, who is also a PhD student in the biomolecular sciences program. Thank you.
Thanks, Anna. So one of the questions that I get asked probably more often than others is what exactly is in these vaccines? So that's what I'm going to try and answer for you guys today. Specifically, I'll be talking about the Moderna and Pfizer vaccines. So, do you have control? Yes, okay. Let's see. Oh, it's a little bit delayed. Okay, there we go. I think Sierra made a great job talking about the mRNA that is in these vaccines. But it's also important how they're absorbed inside your cells. In order for the mRNA to make the spike protein that is going to be used to build your immune response, you need that mRNA to enter your own cells. And that's why the nanoparticles, that also Leslie talked about a little bit, are really important. So what are these nanoparticles made of? Well, they are small containers, like you can see in my little drawing here, and they're made by a phospholipid membrane. It's pretty similar to what you have on, as part of your own cell. And there are also other molecules in there, and I'm gonna list every single one of them. First, the phospholipid membrane. There are some shared ingredients between Pfizer and Moderna. First of all, we have cholesterol, which in the eye of the public is usually thought as, oh, this is really bad, right? We don't want cholesterol. Actually, cholesterol is an essential component of in your own cell. It helps the membrane to remain fluid. That's the same purpose why it has been included in these vaccines. It makes sure that these nanoparticles remains intact. They're not affected by temperature shifts. So it's really important. We also have PEG, which it is in two different forms for the two vaccines, but still basically the same component. It does prevent these nanoparticles to aggregate and also being destroyed by your immune system before the vaccine is actually delivered inside your cell. And also we have DSPC. It's pretty common in a lot of nanoparticles that I've seen in different papers. That is called, it's commonly referred to as a helper lipid, phospholipid. And basically it does help to stabilize the whole structure of the nanoparticle. So again, it doesn't break. And then let's see, Moderna has two unique components for this phospholipid membrane. One of them is called, they call it SM102, which I found the paper that actually kind of discovered why this phospholipid is so important, and where they show that it's actually making the nanoparticle more biodegradable, and so improves tolerability. It doesn't remain in your system, it just it's destroyed naturally. And the other component would be DMG, which I'm not sure exactly what its role is. At least I haven't found much information about it. I know it has been used in nanoparticles before, and I can only guess because it's made out of glycerol, and we use glycerol to protect enzymes and other components and freezing temperature. I imagine that's something to do with storage. It prevents the nanoparticle to break when we freeze them for storage. And then going to the Pfizer vaccine, we have a different phospholipid. It's called lipid that is pretty similar to SM102. They call theirs ALC0315. I haven't found the exact papers where they talk about this molecules, but because of its structure is pretty similar to SM102, I'd expect a similar degree of biodegradability and tolerability. And that covers the whole phospholipid membrane. So I mentioned other molecules, and here they are. Basically, the role of all these molecules is to keep the environment of the mRNA stable, especially when it comes to the acidity of the solution, or pH. That's why both vaccines have buffer solutions. The Moderna has a thromethamine hydrochloride solution, while the Pfizer has a monobasic potassium phosphate and dibasic sodium phosphate dehydrate. So they're the main actor to keep the pH stable. Also, both vaccines have sucrose, which is just a sugar. Moderna has acetic acid, which I know the name sounds scary, but it's actually it's vinegar, and sodium acetate, which is the salt form of acetic acid. So basically just vinegar. And the Pfizer instead has two different kinds of salt. Potassium chloride, it's not exactly your table salt, but it's still being used in medicine and food processing. So we know it's pretty safe for humans. And also the actual table salt, sodium chloride, is the same salt you use in your kitchen. And that covers the whole recipe, well, not the recipe, but the whole list of ingredients for the Pfizer and Moderna vaccines. And that's it. And I want to mention that all these data, you can find it in the FDA fact sheets for the Pfizer and Moderna vaccines, and it's available for everybody. Just need an internet connection, and you can get the same information that I got. So with that, I'm going to introduce Rachel Nichols, which is also a student in the Biomolecular Sciences program.
All right. Hello, everyone. Again, my name is Rachel, and today I will be discussing some global trends and perspectives on vaccination with a special focus on the CDC and WHO data. And Elise, can we go next slide? Oh, we're good right now. All right. So to start, I wanted to address the system that's actually being used to track potential adverse events following vaccination. So this system is called VAERS, or the Vaccine Adverse Event Reporting System. Importantly, this is not new with COVID-19, but was actually launched by the CDC in 1990. This allows vaccinated patients to report any potential symptoms following vaccination directly to the CDC via their cell phones. And in the rare case of a vaccine-related adverse event, they have really easy access to be able to do that. So importantly, like Elise mentioned earlier, she mentioned that something like a broken ankle would be considered an adverse event in a trial. And I wanted to reiterate that a lot of the times these numbers that we see as adverse events can be anything from falling off a 100-foot ladder to, to a car accident. So a lot of times these numbers like, deaths in trials are actually also due to the person just passing away while they were in the trial, unfortunately. So importantly, with the Pfizer and BioNTech vaccine and the Moderna vaccine over on the right, we actually see extrapolated from these larger data pools, only 4.7 and 2.5 anaphylaxis reports per million doses. And those are very, very low numbers. So next slide, please. All right. So here we have a snapshot of the current CDC data indicating what the path to herd immunity actually looks like for this year. So over on the left, we see that at the start of 2021, we had only about 20% of the population with immunity, and that was mostly from previous infection rather than vaccination. Now recall that Tana told you that people who are previously infected tend to not have as high of antibody levels as people who are vaccinated. So during this time, importantly, we're still seeing a pretty sharp upward trend in COVID cases. Now we move to about May. This is a projection, of course, and we expect at the current rate of vaccination, if that continues, we expect to see about 50% of the population immune by May or with neutralizing antibodies. And as a general caveat, you guys, I wanted to say too, that when we refer to herd immunity, what that means is an established about 60 to 70% of the population with COVID-19 neutralizing antibodies. So if we move forward one more step, we can see that by the end of 2021 and about December, we are hoping to reach that 60 to 70% herd immunity. So if we go next, I wanted to just look at what the numbers actually are. We have, our global situation right now is 127 million global cases of COVID-19 with about 2.8 million deaths. And notably, this is April, July, October of 2020, and we can all see that case rates were, increasing pretty consistently into the beginning of 2021, and we see a drop there. That is when we started vaccinating en masse. We also see this death rate was staying pretty consistent throughout 2021, rising with the uptick in cases in the winter. And then when we started vaccinating, very importantly, those death rates were dropping off pretty significantly as well. So if we go next slide, just to summarize, we are hoping with the current situation and the way that vaccination is looking right now with the 3 available vaccines, or the 2 available and the 1 hoping to be available in the next month, that every willing adult in the US can and will be vaccinated, hopefully by about May or June of 2021. And we are well on track to that right now. So to reiterate one more time, we are looking for that 60 to 70% of the population with neutralizing antibodies, with the cell memory in the form of antibodies to recognize the spike protein and prevent infection with COVID-19. So in closing, it's been a really difficult and tumultuous year for all of us. And I wanted to look at a global perspective of case rates of doses administered in these vaccines. So importantly here, we can see that when we're talking about some of these numbers, we have to remember what region we're actually referring to. And right now in the US, we see a much larger rate of vaccination than a lot of the other parts of the world. And I think that's a conversation that needs to be had just because this has to do with availability. And so we always hope to increase that availability globally. So to close, with so much contradicting information, it can be really hard to grasp what's actually going on with the pandemic and some of the numbers. So I hope that this information was really helpful and that you feel like you've gained something. And I will now pass it over to Dr. Tinker for closing remarks. Thanks, guys.
All right. Thank you, Rachel. And thank you, everybody today. You did fantastic. And I'm closing here with this slide because as a scientist who've studied vaccines for many years, when I listened to the FDA approve the first coronavirus vaccine in December of 2020, it felt like we just landed on the moon. I feel like that's probably the way that people felt at that time in 1968. So I think that we still have a lot of work to do, and that's why we really had the students come together to want to share this today. So I'm hoping, I know that we kind of ran out of time to answer questions, but if people want to stick around, we are more than happy to answer some questions. And we do also have an Instagram set up through Boise State where you guys can send in questions and we will answer them on a weekly basis through that. And I also want to hand it off here to Tom at the end so he can just say a little bit about, we also are going to record this for you guys, but let's hand it off to Tom here. Thank you.
Wonderful. Well, thank you so much, Dr. Tinker, and thank you so much to everyone who presented today. This was valuable information. I'm so glad we got to have this. But yeah, I just wanted to let you know we are recording this webinar right now. And so you can find this recording to this session and more in-depth videos of each presentation at the link after April 30th. And so first thing I want to do is just say we would like to also give an extra thank you to Leslie Kendrick, Program Director for our Online Imaging Sciences program, for coordinating this webinar. This has been wonderful. So thank you so much for that. And just wanted to do one little shameless plug. If you are interested ever in completing your bachelor's degree and are a current registered imaging professional, do feel free to reach out and learn more about our amazing imaging sciences program. I'm your success coach, so you'll be in good hands. But yeah, thank you so much to everyone today. Y'all have been wonderful, and this was fantastic. So thank you. All right. Yeah. If anyone has any questions, so I think, Dr. Tinker, did you say we'd be around for just a couple minutes if anyone has any questions?
Yes, we can stick around for a few minutes if anyone has questions.
Okay.
I have a question if it's okay to ask.
Yes,
Hello,
sure.
all. And I guess this is for the entire panel and for you as well, Dr. Tinker. For individuals who just want to start just like a baseline foundation of gathering information if they're unsure about moving forward with getting the vaccine, what is kind of an easy peasy just go-to place that you or panelists would direct people to go to start information gathering? I know we hear a lot of conflicting things, and so what's a great place for somebody to just jump in and start learning?
I'll just say real quick that the CDC, I believe, is really, they actually do have really pretty approachable, easy information there. So that's what I would do, but I'm going to open it up and let others answer that question.
Dr. Tinker, I was going to agree with you. Start with the CDC. That's where I found my little infograph, and there was a little bit more information on that infograph, but just throughout the CDC.gov website in general, there's different tiers of information as well.
I also just posted in the chat a link that I use rather frequently through Johns Hopkins School of Medicine, and they have a pretty up-to-date information on the vaccines that's pretty accessible.
Thank you.
I would say also the FDA fact sheets that John Luca was talking about, those are great resources, and they've been a resource that I've given to other people because it gives them an idea for comparison of expected signs and symptoms that they might experience after the vaccination, and also gives information about what's inside of each one, so you can compare across the board if you're concerned about what are the contents. So I thought they did a really nice job of laying out all the information that's related to the actual contents and what you can expect with a vaccination. So I would refer you to the FDA website, fda.gov website as well.
Hi, this is Chase Biefelt at KTVB. My question would probably be for Tana, I think, because it's about the Novavax vaccine. So we talked about, or you talked about earlier, not Tana, but somebody was talking about mRNA vaccine and how it kind of gives you the information your body needs to defeat the virus. But we know that the Novavax vaccine is entirely different in the way it's kind of presented to your body. And you explained a little bit of it, but you know, I'm not big on science, and I have to, like a student, I have to ask further questions and follow up. So do you think there's any way you can kind of like dumb that down a little bit and explain how that is presented to your body differently from mRNA?
Sure. So mRNA is basically the step before the spike protein. As we mentioned before, the mRNA is the instructions for your cell to make that protein. And so the difference, I guess, in my view, is that Novavax just makes that spike protein already before, like, so your own body doesn't have to make the spike protein. We have already made it in these moth cells. And then we take it and we just put it onto this nanoparticle so that your immune system has something to attack. And so the mRNA vaccines, they basically give your cells, I'm going to pretend, this is a bad figure, but they basically package that mRNA up and give them to your cells, and your cells take them and they use those instructions to make this protein. So basically the mRNA vaccines are just a little extra step as compared to the Novavax ones. Does that help explain a little bit better?
I think so.
Can I add something to that explanation?
Yeah. Yes, please.
I wanted to recap. Dr. Tinker had mentioned the DNA to RNA to protein situation. And we learned this in every science class we've ever taken. It's the central dogma of biology. So you get DNA to RNA to protein. DNA is transcribed into an RNA instruction, which is translated into the protein of interest. So what happens here, we've got some DNA vaccines out there, right? And then we've got some mRNA vaccines. And then now we have the Novavax, which is step 3. It's the protein itself rather than the instructions. I hope that was supplemental.
I can just
clarify, I think so what's has not been explicitly stated is the Novavax vaccine is actually the spike protein. So what we are injecting into patients' arms is actually the spike protein itself.
Okay, so I guess my question now would be, that makes a little more sense as to how it's introduced, but how does it actually affect your, I guess, your body and what is happening with that? Could it have possibly different effects?
Let Dr. Tinker talk about the immunology part, but what your immune system sees and reacts to is the exact same. So with the mRNA, your body makes the spike protein, and that spike protein is what your immune system reacts to. And in this case, we're just putting the spike protein straight into your body so your immune system sees that. But in all the cases with all of these vaccines, the end result is that your immune system sees a spike protein and can react to it.
So is the Novavax vaccine technically more efficient than the other ones?
There are a lot of pros and cons to all of the different vaccine platforms. There are problems with putting a protein straight into your body too. Your body will recognize it as foreign and start to break it down quickly. So one of the advantages of the mRNA vaccines is that they have that little liposome, they have that little capsule that they come into your body in, and those can help that to evade your immune system right off the bat. So the vaccine is kind of protected from your immune system right off the bat, whereas with the Novavax one, your immune system directly is interacting with it and attacking it and breaking it down. Dr. Tinker, you can probably add something better.
Well, I just wanted to add that we have a lot of purified protein vaccines already. So that's something that we've done before, and we know that it works. We know they're stable. We know they're really safe. I think vaccinologists were a little bit concerned that mRNA vaccines were going to be unstable. They weren't concerned about safety. They were concerned that they were going to be unstable and just not work because of that. But as it turns out, they work fantastic, which was a little bit of a surprise, but a good one. Anyone else have anything they want to add?
Or keep asking questions.
Or keep asking.
We're totally on this bandwagon.
I'd like to add one more thing that wasn't stated maybe explicitly also. And that's that when we're introducing something and we want an immune response from it, that is a sequence specific immune response. So these memory B and T cells, they will recognize the sequence of the spike protein. It's like if you were a hunter and you were going deer hunting, but you'd never seen a deer in your whole entire life. The mRNA would be like the picture of the exact deer you're looking for. Was that a good analogy, you guys? So we're giving the sequence specific instructions for the spike protein.
So it's all a journey. It's just you're getting there in a different way with the different vaccine. Okay.
But we are all ending up with the spike protein, showing it to the spike protein to the immune system.
Yeah. Elise, I was going to say that one picture that I used at the bottom that showed just the simple picture of the spike protein.
Oh, this
it
guy?
Shows, no, right there, yeah. So Chase, what it does is you see that spike protein, the purple on the bottom. That is what we're all trying to get our immune system, which is that human cell. What you see there, there's the angiotensin converting enzyme 2, which they call the ACE2. You may have heard about the ACE2 receptor that everybody talks about, the spike protein adhering to, and our body recognizing the spike protein. And so that we're just have different ways of either developing the spike protein or putting the spike protein directly into the individual so that it will be recognized before it attaches onto that ACE enzyme and then moves into the human cell. So that's all we're trying to do, is get our body to recognize it before it attaches and causes an infection. I don't know if that helps.
This all definitely helps. Thank
Just
you.
come take a biology class with us.
Do we have any other questions?
All questions are good questions, by the way.
Is there anybody in the audience that has gotten vaccine, has received their vaccine just yet?
Mhm.
Yay.
Is there anybody that's a little leery about getting their vaccine that we could answer any questions about?
What are some of the other major questions that you guys get from people that you can think of?
A question that I have gotten a lot about the human trials is who signs up for trials, and are the doctors who make the vaccines or the scientists who make the vaccines testing it on themselves? And so the people who sign up for trials are really, I know I said it in my thing, but it's like really just normal people from the community. The scientists who make the vaccines shouldn't ethically test it on themselves because they would be biased. They would already have a thing they expected to happen. So they wouldn't have good unbiased data. And that's why they don't tell you when you're in a trial, they don't tell you. You either get the vaccine or you get a placebo, which is not the real vaccine. It's a saline shot. So that's the reason that they don't let scientists be in their own studies, is so that we don't get biased data out the other end.
Yeah, I was actually in the Novavax vaccine trial. I was in that one a little while ago, but I did get the Pfizer. I was able to unblind. Oh no, I had the placebo, so I went and got the Pfizer. But yeah, no, it was a wonderful thing, and being able to help science is something I've always wanted to do. So I think if you have the opportunity to help save lives, go for it. It's a wonderful opportunity.
Yeah, we have a lot of the trials going on in the Treasure Valley. There's at least three sites that I know of that are doing vaccine trials still in the late phase and the pregnancy studies, and it's happening here.
So one more question I have that just kind of popped up. A lot of vaccine doubters who, have never gotten a vaccine, we've seen them, say they don't know the long-term effects of these, especially, and they have doubts, especially since, it doesn't have that full licensure from the FDA yet. It was just an emergency use authorization. So is there any way to tell the long-term effects right now, or is there any way that those have been projected? Is there any way to kind of have a picture of that as of right now with these COVID-19 vaccines?
So I suspect that people are going to jump in on this question, but I'm going to start. So I think it's important to point out that, yes, it's an emergency use authorization, but the difference between that and licensure is not a skimping on safety in any way. So the safety, even when we license a vaccine, we still look at safety for about 2 months before it gets licensed. So they didn't skimp on the safety parts of authorizing these. It was just looking at efficacy, which is very important, of course. And so it's really important we keep the trials going. But there was no skimping on looking at safety before these were put into people's arms. And so our licensed vaccines, yes, now they've been licensed for many years. We can look back and say there's no safety issues for long term. But we kind of look at long-term safety. We kind of think about biologically what are the possibilities that could happen long-term. And there really are very low concerns about all of these vaccines biologically, looking out long-term. But it's something that we still just have to kind of keep studying as we go. So It's just that the risk is likely really, really low if there is one at all.
I think we can probably like Gianluca works a lot with RNA, so he might be able to jump in on this, but the vaccines themselves, like the reason that there's low risk, we can maybe add some actual background of why there's low risk. So like the mRNA vaccines are made up of, like Gianluca said, of lipids and the mRNA. And both of those things are already in your body. Like all of those ingredients are, your body already knows how to break them down. So we're not introducing something totally foreign that's going to have, that your body's never had to figure out how to deal with before. Your body already knows how to break down lipids. It already knows it wants to break down RNA really fast. And that's part of the problem that they've had. That's why it took decades to get an mRNA vaccine. We had to figure out how to keep it stable for long enough to have an effect. So that, and then like the Novavax is a protein, and the protein will get broken down. Your body already breaks down proteins all the time, every day, every second, thousands of proteins every second. So the protein will get broken down. So those are just some, maybe to add some of the biological, like, how do we know that they're, how do we know there's not a long-term risk? Well, we don't know there's not a long-term risk, but we can say, we know that they're going to get broken down quickly, and that they're not a foreign thing.
If I can add to that, you mentioned earlier, we have like different approaches for the vaccines. We have the DNA ones, we have the RNA ones, and the protein ones. Well, the RNA, it's so much more susceptible to degradation compared to DNA and proteins that it should be really safe compared to all the vaccines we've had in the past. So this is why these vaccines are so groundbreaking. We're using nanoparticles that are biodegradable. We're using RNA vaccines. So yeah, they're pretty awesome.
And to add one more thing is that, the like somebody mentioned early on, the RNA vaccines themselves are not new. And we do have some long-term data from the SARS epidemic or pandemic of 2002, and the MERS pandemic. So with this technology, we have some long-term data dating back about 18 years.
And also, I just wanted to state that these phase 1 and 2 trials have been going on for a little bit over a year now, especially with Pfizer and Moderna. And so they don't just stop monitoring safety after a few months. I know in phase 3, in the Novavax phase 3 case, they monitor people for 2 years after that. So especially since the sheer amount of people we've had vaccinated already, but just looking at, it's been over a year and there hasn't been any serious effects so far. So how long-term is long-term?
It looks like there is a question in the chat. It says, how can we address if a vaccine is affected by someone's autoimmune condition? Are there many people with Hashimoto's and vitiligo? And I hope I said that correctly.
Dr. Tinker, do you have an answer for this one?
I can speculate. Autoimmune disorders should not have any effect on the efficacy of the vaccine. So this is, autoimmune disorders are not necessarily, it's more that your own immune system is kind of overactive and instead of being underactive. So in that case, you still would create an immune response against the vaccine. It's probably going to still be very effective. And it should not have an effect on your actual, it shouldn't make it worse. That being said, we do have adjuvants present in these vaccines. There is a possibility, I think, that it could make it worse, but I don't think any of our clinical trials or real world studies right now are indicating that it does, but it still is probably something you should talk to your doctor about. Does anyone have anything they want to add to that?
I did want to.
Oh, go ahead, Hannah.
I did want to add that if you are on any immune suppressant medications, it could make your immune system weaker, so that maybe you don't form as great a response to the vaccine as you would. So that would be something that you would want to ask your doctor about as well.
I was going to say as well that Dana, like you mentioned, vitiligo, a similar skin condition, psoriasis, for example, has a dysregulation of very specific receptors or immune cells oftentimes. And in some of these really common autoimmune diseases, those cells that are dysregulating are oftentimes not your memory B and T cells. If you had a significant dysregulation of that, you'd probably see a lot more significant disease. But the example of psoriasis is that, when we're even immune medications for that are targeting like the TNF alpha receptor only. So it has nothing to do really with the B and T cells, but something else more specific. And that's why we're able to still vaccinate a lot of those patients with mild autoimmune conditions.
I have just one thing to add. I actually have family members who have Hashimoto's, so I looked into it a little bit early on when vaccination was coming out, as well as Down syndrome as well, which is considered an autoimmune disorder as well. And for both of those, they are recommending vaccination by both the CDC and the WHO because there has been no negative impacts for either of those conditions.
All right. Any other questions? All right. Well, again, thank you all so much for your time today and presenting this wonderful information. As we noted, this presentation has been recorded and we will have stuff available on April 30th. Do feel free to reach out if you do have any questions. And does anyone have any other last minute thing they want to add before we go today?
I just wanted to say thank you for bearing with me and explaining some of that stuff. I do feel like I'm a student again and asking questions. It feels really good to be able to ask multiple follow-up questions as a journalist. So thank you so much for being patient and explaining some of that stuff.
I would say we all have spent a lot of time at this point researching and figuring this stuff out ourselves. And it's not easy, and a lot of us have put a lot of work into it. So for me at least, I won't offer everybody, but anybody who has questions is welcome to ask me, because if I don't know the answer, I should figure it out anyways. But all of us have put a lot of energy into this. So we like when people ask questions because we have done the work on the scientific end. So if we can spread it out and make some connections, build some bridges between the crazy academic scientists, all these 1,000-page papers, if we can take that and share it with other people who don't have the background, that's what we're trying to do. So
yeah, at least I would piggyback off of that. I would say that the more you learn, the more you realize that the more you try to learn more, so you know more, the more you realize you don't know. And so whenever someone asks us a question that we don't know the answer to, it's an opportunity for us to learn too. So it's exciting, actually. And we look forward to having questions posed to us that we can find out the answers if we don't know.
Well, thank you all of the students. You guys did awesome. And thank you all for coming. It's been great.
Thank you all. Well, everyone have a great day and take care.